Welcome to our channel. Today we are going to discuss amino acids in humans. Let's get started. Amino acids are organic compounds that serve as the building blocks of proteins. They play crucial roles in various physiological and biochemical processes in the human body. Dietary sources of amino acids: animal-based, meat, fish, eggs, dairy products; plant-based, legumes, nuts, seeds, quinoa, soy products. Amino acid pool. The body maintains a dynamic amino acid pool in the blood and tissues, which is used for protein turnover, gluconeogenesis, synthesis of non-protein compounds. General structure of amino acids. The basic structure of an amino acid can be summarized as HNCH,R,COOH, where alpha carbon is the central carbon atom to which all functional groups are attached. Amino group, NH, is basic functional group. Carboxyl group, COOH, is acidic functional group. Hydrogen atom attached to the alpha carbon. Side chain, R group, unique to each amino acid, determines its chemical properties. Chirality of amino acids. Most amino acids, except glycine, have a chiral alpha carbon, leading to two possible isomers, L and D. Forms. L amino acids, found in proteins and predominant in nature. D amino acids, found in bacterial cell walls and some antibiotics. Glycine, non-chiral because its R group is a hydrogen atom. Ionization properties. Amino acids exist as zwitterions at physiological pH 7.4. Zwitterion means molecule that contains an equal number of positively and negatively charged functional groups. The amino group is protonated, NH. The carboxyl group is deprotonated, COO. This dual charge contributes to the solubility and buffering capacity of amino acids. Isoelectric point is the pH at which an amino acid carries no net charge. Side chain, R group, variability. The R group determines chemical properties, polarity, charge, hydrophobicity, structural role in proteins, reactivity and interactions with other molecules. The side chain's properties form the basis for classifying amino acids, for example, nonpolar, polar, acidic, basic. Peptide bond formation. Peptide bond, formed by a condensation reaction between the carboxyl group of one amino acid and the amino group of another. The resulting bond is planar and rigid due to partial double bond character. Chains of amino acids form peptides, polypeptides, and ultimately proteins. Functional groups. A. Amino group, NH, basic group, capable of accepting protons. Participates in hydrogen bonding and ionization. B. Carboxyl group, COOH, acidic group, donates protons at physiological pH. Key in forming peptide bonds. C. Hydrogen atom, H, non-reactive but essential for structural balance. D. Side chain, R group, determines chemical reactivity, solubility, and interaction with other molecules. Specialized structural features in some amino acids. Proline, cyclic structure. The amino group is part of the side chain, making it a secondary amine. Rigid, induces kinks or bends in protein chains. Cystine, contains a thiol group, SH, that forms disulfide bonds. Stabilizes tertiary and quaternary protein structures. Histidine, imidazole group has a pKa near physiological pH, allowing it to act as a proton donor or acceptor. Aromatic amino acids like phenylalanine, tyrosine, tryptophan contain aromatic rings. Absorb UV light, contributing to protein quantification. Tautomeric forms. Amino acids can exhibit tautomeric behavior under different tautomers are structural isomers, constitutional isomers, of chemical compounds that readily interconvert. The chemical reaction interconverting the two is called tautomerization. This conversion commonly results from the relocation of a hydrogen atom within the compound. The phenomenon of tautomerization is called tautomerism, also called desmotropism. Tautomerism is, for example, relevant to the behavior of amino acids and nucleic acids, two of the fundamental building blocks of life. Physical properties. Solubility. Amino acids are soluble in water due to ionization but vary based on the R group. Melting points. High melting points due to ionic interactions in the solid state. UV absorption. Aromatic amino acids absorb UV light, maximum at 280 nanometers. Biological relevance of structural features. Enzymatic catalysis. Side chains form active sites in enzymes. Protein folding. Hydrophobic and hydrophilic interactions drive protein structure. Signal transduction. Phosphorylation of hydroxyl-containing amino acids, SIR, THR, TIA, regulates signaling pathways. Buffering. Ionizable groups help maintain pH homeostasis. Classification of amino acids. Amino acids are classified based on various properties such as their chemical structure, polarity, charge, and metabolic behavior. These classifications help in understanding their roles in protein structure, enzymatic functions, and metabolic pathways. 1. Classification based on polarity and charge of side chains. Here the figure shows basic positively charged amino acids, in which side chains contain amine groups that accept protons at physiological pH, becoming positively charged. For examples, lysine, arginine, and histidine. Right side shows acidic, negatively charged amino acids, in which side chains contain carboxylic acid groups that lose a proton at physiological pH, becoming negatively charged. For examples, aspartic acid, glutamic acid. Polar, hydrophilic amino acids, in which side chains interact with water through hydrogen bonds, found on the surface of proteins, interacting with the aqueous environment. Examples of uncharged polar are serine, threonine, cysteine, tyrosine, asparagine, glutamine. Non-polar, hydrophobic amino acids, in which side chains are hydrophobic, containing primarily hydrocarbons, found in the interior of proteins, away from the aqueous environment. Examples are aliphatic, glycine, alanine, valine, leucine, isoleucine, methionine. Cyclic, proline. Aromatic, phenylalanine, tyrosine, tryptophan. Classification based on nutritional requirements. Essential amino acids cannot be synthesized by the human body, must be obtained through the diet. Its examples are valine, leucine, isoleucine, lysine, methionine, phenylalanine, threonine, tryptophan. Histidine. Non-essential amino acids can be synthesized by the human body. Its examples are alanine, asparagine, aspartate, glutamate, serine. Conditionally essential amino acids required in the diet under specific conditions like illness, stress. Its examples are arginine, cysteine, glutamine, tyrosine, glycine, proline. Special categories. Branched chain amino acids. BCAAs. Essential amino acids with branched aliphatic side chains. Examples: valine, leucine. Isoleucine. Amino acids with ionizable side chains. Can gain or lose protons depending on pH. Influencing protein charge and activity. Examples: aspartate, glutamate, lysine, arginine. Histidine. Amino acids. Proline is categorized as an amino acid due to its unique cyclic structure, where the amino group is part of the side chain. Metabolism of amino acids. Biosynthetic pathways. A. Synthesis of non-essential amino acids. Glutamate. Alpha-ketoglutarate, an intermediate of the citric acid cycle, is converted to glutamate by glutamate dehydrogenase, GDH, through reductive amination, and by transaminases, which transfer an amino group from other amino acids to alpha-ketoglutarate. Alanine synthesis occur through transamination of pyruvate by enzyme alanine. Transaminase. Aspartate synthesis occur through transamination of oxaloacetate by enzyme aspartate transaminase. Glutamine synthesis. It is synthesized from glutamate by the addition of ammonium ion, NH4. Glutamine synthetase catalyzes the reaction in an ATP-dependent manner. Asparagine synthesis. Asparagine is synthesized from aspartate through an amide transfer reaction. Asparagine synthetase catalyzes the ATP-dependent transfer of an amide group from glutamine to aspartate. Serine synthesis. Serine is synthesized from 3-phosphoglycerate, 3-PG, an intermediate of glycolysis, via 3 enzymatic steps. First step is oxidation, in which D3-phosphoglycerate produce 3-phosphohydroxypyruvate. Enzyme involved is phosphoglycerate dehydrogenase, and NAD is reduced to NADH. Second step is transamination, in which 3-phosphohydroxypyruvate produce 3-phosphosarine. Enzyme involved is phosphosarine. Aminotransferase. Glutamate donates an amino group, forming alpha-ketoglutarate. Third step is dephosphorylation, in which 3-phosphosarine yields serine by enzyme phosphosarine. Phosphatase. Glycine synthesis. Serine tetrahydrofolate react to form glycine. N5, N10 methylene THFHO. Enzyme involved is serine hydroxymethyl transferase. Glycine also synthesized from choline. Proline synthesis. Proline is synthesized from glutamate through a multi-step enzymatic process. First step is glutamate to glutamate gamma semialdehyde by enzyme pyroline 5-carboxylate synthetase. Glutamate is phosphorylated and reduced to glutamate gamma semialdehyde. Second step is glutamate gamma semialdehyde to pyroline 5-carboxylate. RIT involves spontaneous cyclization of glutamate gamma semialdehyde forms P5C. Last step is P5C2. Proline by enzyme PYRRO-LINE5-carboxylate reductase. P5C is reduced to proline using NADH or NADPH. Catabolism of amino acids. First step is transamination, which involves transfer of an amino group from an amino acid to an alpha-keto acid by enzyme transaminases. Aminotransferases. Cofactor involved is pyridoxal phosphate, PLP. derived from vitamin B. Here is an example. Alanine alpha-ketoglutarate form pyruvate glutamate. Second step is deamination, which involve removal of an amino group from an amino acid, forming ammonia. Its types are oxidative deamination, catalyzed by glutamate dehydrogenase. Non-oxidative deamination occurs in serine and threonine. Here amino group removed from alanine. 3. Decarboxylation is the removal of the carboxyl group, COH, from amino acids to produce amines. For examples, decarboxylation of histidine form histamine. Decarboxylation of tyrosine form tyramine. Tyramine elevates blood pressure. 4. Urea cycle, ammonia detoxification. This process converts toxic ammonia into urea for excretion. The initial step of the urea cycle involves the conversion of toxic ammonia, NH3, to a less toxic compound called carbamoyl phosphate. This reaction is catalyzed by the enzyme carbamoyl phosphate synthetase 1, CPS1, which requires 2 ATP molecules and bicarbonate, HCO3, as substrates. CPS1 is primarily located in the mitochondria of hepatocytes. In the mitochondrial matrix, carbamoyl phosphate is created and then reacts with ornithine to create citrulline. Ornithine transcarbamylase is an enzyme that promotes this process. The remaining processes of the urea cycle subsequently proceed in the cytoplasm. Once citrulline has been transferred over the inner mitochondrial membrane, argininosuccinate synthetase catalyzes the process in the cytoplasm where citrulline and aspartate combine to generate argininosuccinate. Argininosuccinate liase further cleaves argininosuccinate to provide fumarate and arginine. After being hydrolyzed by arginase, arginine produces urea and ornithine, both of which can re-enter the mitochondrial matrix and take part in the urea cycle once more. Glucogenic amino acids, converted into intermediates of gluconeogenesis, for example pyruvate, oxaloacetate. Examples: alanine, glutamate. Serine. Ketogenic amino acids, converted into acetyl-CoA or acetoacetate for ketogenesis. Examples: leucine, lysine. Both glucogenic and ketogenic, phenylalanine, tyrosine. Isoleucine, tryptophan. Disorders of amino acid metabolism. Phenylketonuria, PKU, caused by deficiency of phenylalanine hydroxylase. It leads to accumulation of phenylalanine, leading to neurological damage. It can manage by taking low phenylalanine diet. Maple syrup urine disease, MSUD, caused by deficiency of branched-chain alpha-ketoacid dehydrogenase. It leads to accumulation of branched-chain amino acids, LU, ILE, valine. Management is dietary restriction of branched-chain amino acids. Alkaptonuria, caused by deficiency of homogentisate oxidase. It leads to accumulation of homogentisic acid, leading to dark urine and joint issues. Homocystinuria, caused by deficiency of cystathionine beta synthase. It leads to high homocysteine levels, leading to cardiovascular and neurological issues. Functions of amino acids. Protein synthesis. Fundamental building blocks of proteins. Enzyme production. Act as precursors for enzyme synthesis. Hormone synthesis. Tryptophan yields serotonin. Tyrosine yields thyroid hormones, epinephrine, norepinephrine. Energy production. Amino acids can be metabolized to provide energy. Cell signaling. Regulate cellular processes and communication. Immune response. Essential for producing antibodies. Nitrogen balance. Maintain nitrogen equilibrium in the body. Detoxification. Assist in the elimination of harmful substances. Thanks for watching.